Is that started recording?
Yeah.
Okay.
So the recorded. Yeah.
All right. That's good. Thank you.
Yeah.
All right. I think we can get it started. Sorry about the late. So this is the course that is being collected in the School of Informatics and School of Medicine. And I started to teach this course from 2011, so it has been a couple of years. And my name is Yunlong Liu. I'm from Department of Medical and Molecular Genetics Department. I'm also part of the Center for Computational Biology and Bioinformatics, which locates on the 5th floor of this building. And also, I'm also directing the Center for Medical Genomics, which runs the sequencing core facility for Central Indiana. And the core locates in the R3 building. I assume that most of you know the campus, but that is in R3 building on the 4th floor. And the reason I mention this is that there will be a lecture that we do not take classes, but we go to the sequencing core as groups to tour the facility and giving you the opportunity to, to see what the machine really look like, okay? And today is the first lecture, and which I will briefly go through individual lectures, what they are trying to achieve. I think this moves. All right, that's fine. All right. So some of the logistics before I get him started. You know the course place, and we'll be here and HS 1110, and Fridays from 3 to 6 p.m. I know this is usually the time that you have beer, but you probably want to do that after the course. This one doesn't really move things. I don't know. I have to press. Okay. So we have two course coordinators, which is myself and also Sean Courtney. Can you stand up? Yeah. So that is Sean Courtney, which I will introduce the responsibility a little bit. So what I'm trying to do is I will skip most of the activities that I need to do. That's okay. I will just press the buttons. Is that? Yeah, you plugged in the wrong port for me. So now it works. That's why I didn't. All right. So if you have any questions so I didn't set up an office hour for this semester, and simply because my schedule is extremely tight. But I think Sean can take that responsibility. Can we negotiate now? Is that Friday morning is a good time?
Absolutely.
Yeah, Friday morning, 9 to 11. Perfect. 9 to 11 will be office hours. So that will be in his office. Okay, it's on the 5th floor. And what's your room number?
5025.
5025. You need to write the notes down. Okay. So we will have a couple of people who gave the lectures throughout the semester, and I will take probably a little bit more than half of all the courses. And Dr. Chi Zhang, if you stand up, and he will give 3 lectures. That's fine. You can. He gave 3 lectures on the RNA sequencing part, which I will introduce the syllabus a little bit. And Dr. Jun Wan, where's Jun? Yeah. And towards the end of the course, and he will be talking about the ChIP-seq and the other technology related to the gene regulation. Sean will be in charge of the office hours answering questions, but at the same time, he has a very important component which will go through the hands-on section of the next generation sequencing part of it. So, which I will talk about in a little bit more detail. And the last person of the staff is Dr. Matteo Vata. And he is actually a clinical geneticist, and he does this next generation sequencing really in the clinical setting. And I often invite them back to come give half a lecture and tell us that the real application, what are the considerations in addition to the bioinformatics concerns. So most of them are in the regulatory component of it. Okay, and this is my office and my email address and the phone number. We will do not have the textbook, so you can save that $100. And for the reason that I cannot find any textbook that fits this. Okay. So I want to briefly describe about the course objectives, and which is important actually, because when I talk to the students, and a lot of people that think that after you take this course, you are being the expert of next-generation sequencing data analysis. I'm going to tell you that's not going to happen. Okay. So what we really to achieve here is I want to introduce the basic principles of next-generation sequencing technology. And so include several different components. The basic biological applications that is associated to this technology. The basics in the data processing part. So what are the different steps that I need to go through to really navigate the data? And the statistical and informatics series in data analysis. And this part sounds like a pretty scary, right? Especially for people who has the biology background. But actually what I expect you to understand absolutely not about those complex equations. I will get into a little bit in the course, throughout the course, but I do not expect you understand everything or you can derive everything. What I need you to know is what are the advantages and the limitations of each individual algorithm. And because they are all based on different assumptions, what are the assumptions of different methodologies? That is absolutely critical, okay? And also towards the end, I also talk about the biological interpretations of the results. And many people, especially for the students in the School of Medicine, and send me email asking that I don't know too much about Linux, I don't know how to do the programming. Is that okay? And I would say that it is okay for this course. And the main focus, the purpose of this course is actually for the biologist to understand what this technology can do. And what are the methodologies behind that. So if you are starting a project and you have some of the informatics collaborators that are working with you, and then you need to ask the educated questions. And you need to make sure that they explain the results well back to you. And I want to make sure that I lay the foundation so that you have the knowledge for that part. For the School of Informatics students, I would say the other way around. So you need to understand the basics before you do any programming or developing any algorithm. So I think this will be beneficial for both two sides, okay? And for the course evaluation, and we will have about 10% of the final credit will be given by the in-class quiz. And the reason I want to have that is you start to review each lectures after the lecture, not just before the exam. And that's number one. Number two is I want to make sure for the students who register the course, you are present in the classroom. So, and if you say, I'm going to be out of town for a conference or for a family event, absolutely okay. Tell me before the class. And as long as you email me, I see the records, and I will not penalize you, and it's just because of you are not there. Thank you very much. All right, I paid for it, okay? That is my card. Make sure I put it back. And we will have 10% of the grade to the homeworks, and which Sean is going to go through, and we will have a few throughout the course. And we will have project or literature review. This will count for 30% of the final grade. And there are some logistics you can go through. But the general idea is you are expected to give a presentation, which is about 10% of the final your score, and the final report is another 20%. And we will have midterm exam and on the October the 13th, and which will account for 20%, and the final exam will be 30%. And final exam will be comprehensive, okay, meaning that throughout the whole semester. In the past, I didn't have, at least for the first 4 times I was teaching this, there was no midterm exam. There was only one final. I thought that is good. And but the students really complained after final exam. Oh, it can be that difficult. And so I said, okay, let's have a midterm so you know what to come in the final, right? Any questions so far? So we all agree on this, okay? All right. So let's first let me start to go through the syllabus a little bit. Oh, by the way, how many of you have already the access to the Canvas system? Okay, let me ask you this question. How many of you who registered for the course do not have the access to the Canvas system? It's not showing up?
No, the course isn't showing up on my Canvas.
When did you register?
Beginning of the summer.
So you just checked it wasn't showing up?
Yeah, I checked it at the beginning of the class, and hers showed up, but mine didn't.
Oh, yeah. If you have a question like this, and contacting me will be absolutely useless. Okay. And the register office or register office probably, and those people will help you to figure out. Maybe are registering through the School of Informatics.
Yeah, and I'm on the class list.
Right, and probably Robin is the one that you want to talk to. Oh. Right. This is a really interesting system. At one point, will break my back, I think. Right, hold on. Oh, we'll make it work. All right, that's perfect. So today is the first lecture, and the second lecture will be focusing on the data processing part. So what I'm going to talk about is going to be anything that is not related to a specific biological applications, including the sequencing alignment, the data format, and what are you expecting in when you deal with the data analysis. And then Sean is going to give an introduction to the IU supercomputer environment. So you know that when you run these applications and how to do that, or have an idea where to find the help. And then on September the 8th, and we are going to have a tour to the Center for Medical Genomics. And we will think through the logistics because we are having a big party if everybody goes and the machine is going to be damaged. So we will have a group of maybe 5 people as a group and for about 10 minutes or so, and then we kind of work out the schedule for that. And after that, from September the 15th, we are going to have 3 lectures talking about the DNA sequencing. And this is for genomic DNA sequencing, which we'll get into. For the first lecture will be more on the biology component of it. For the second one, it's the informatics part. What are the qualities, how to do the calculation, how to do the variant calling, how to do the refined calling, and this type of business that you need to know. And then after that, we will have after you identify genetic variants and how do we prioritize making story out of the variants being identified. And then you can see that in the third lecture, the second half will be Dr. Matteo Vata talking about using whole exome sequencing or other target-based sequencing in the clinical setting. And his expertise is in the cardiovascular disease. And on October the 6th, we are going to also talk about DNA sequencing, but the first half is going to be metagenomics and microbiome sequencing. And this is not really my area, but I asked another faculty member in the center help me to prepare for the slides, and then last year she did that, and it worked great. And then before the lecture, she used about 2 hours and taught me about it. And then I came out and explained what I learned. I found that's really rewarding, and it's very important. And there are two things. One is I learned, and that is important. And the second thing is I can use the most simplified language to you and tell you what everything you need to know about it. And because that's to the extent I know about it. And October 13th will be important, and that will be a midterm exam. And then we will have 3 lectures talking about RNA sequencing and data analysis. And also starting from the basic biological questions and the gene expression analysis. And then the second lecture is going to be related to the alternative splicing analysis and the transcriptome assembly. And these lectures will be taught by Dr. Chi Zhang. And then we will have a hands-on section in terms of the distributed RNA sequencing pipeline. And after that, we'll still have 1 more lecture to give the advanced topics of the RNA sequencing. And this is the first time we put in the single-cell RNA sequencing and into the picture. And so I think I really look forward to this. And then after that, we'll have 2 lectures by Dr. Jun Wan talking about using the next generation sequencing for gene regulation. That will include the transcription factor, DNase, ATAC-seq, high-seq, high-C, and the CLIP-seq, many very interesting assays. And then the methodology that related to the data analysis. And then towards the end, in the second lecture, we'll talk about the application in the epigenetics studies. And then Thanksgiving and December 1st will be the student presentation. And if we have time, we'll do the course review. And then December 8th will be the final exam. Okay. So any questions so far? No question. That's great.
One question.
Yes.
For the hands-on instruction, would you like us to bring our own computers or?
I think the way that we probably will do this is Sean is going to go through the series and have a list of operations that you need to do, and he will be giving a live demo to a certain extent, but you got to practice, go back to practice. And the reason for that is for many of these analysis, you are not expecting to get a result instantly. And sometimes it takes hours, sometimes take days, okay? But one thing I'm trying to, which we'll talk about a little bit, I'm trying to negotiate with the Illumina because they have an online platform called Base Space. It's a cloud-based computing, and it's free for the users, unless you, I mean, as long as your data is less than 1 terabyte, and they have many different applications built in. And so I'm trying to arrange a webinar with them and open to the class, so that even if you are really very against the Unix system, which I will not blame you if you do, and you are not familiar with IU supercomputer system, if you hate IU supercomputer system for some reason, and which I will not blame you if you do, and you can still use the cloud computing to do certain things. But I think that's a good exposure. That is, in general, the trend to do this type of analysis, regardless of what we develop now, and eventually will go on the cloud. It will be a good practice. So I'm arranging with Illumina to have a webinar, but that it will be out of class. I will send out the announcement once it becomes a reality. Okay, so now let's come to the lectures. All right. So today I will go through things quickly because I really have a lot of things to talk about. And the first part is I want to give a general idea, general concept, what is next generation sequencing technology. I know many of you heard about it, and sometimes even deal with about it. And if you go to the conference, it's hard to escape, right? But I want to give you a more formal definition from my mind of what that is. And I will go through a few platforms that is we can consider is mainstream, especially the Illumina sequencing. And for this part, what I want to tell you is not only tell you this thing sequence really fast, but tell you why it sequenced that fast. So what are the biochemistry behind it, okay? And I want to make sure that you understand enough to appreciate this is not a black box. This is some smart people design a very smart way to do the sequencing. And it's really fascinating. And I will go through some of the biological applications and present some challenge towards the end. So first, what is next generation sequencing technology? Okay, so what is the sequencing? I mean, we know what is sequencing, right? We know that as a human, we have 3 billion letters in our genome, and the chromosome is kind of in the nucleus, and they pack in this thing called chromosome. But if you go detail level, it looks like this, right? It's double helix and has 3 billion letters. And we have 1 copy from father, 1 copy from mother, and approximately there are 20,000 to 30,000 genes throughout the genome, and there are many biological signals that are also very important, even though when they are not coding for the proteins. So those are basic biologies. So those are, I mean, sequencing is just trying to figure out what is the sequences. And why are we going to do the sequence? I will go through this quickly, right? This is the molecular biology central dogma. So you see this is DNA to RNA is transcription, RNA to protein is translation. And the protein, in most of cases, is achieving its functionality. So the nucleotide order determines amino acid order and by extension, the protein structure and functions. Meaning that the DNA sequence and eventually will be converted to the protein amino acid sequences and eventually achieve its functionality. So it's very important for us to know the sequences of our DNA or RNA at different levels, okay? And we also know that changes in the DNA sequence sometimes is problematic and sometimes it can cause disease. And these are just some simple facts that we all know that for the protein, altered non-functional, altered or non-functional protein will cause a harmful in the living organism. So if there's a gene, there's a SNP that causes amino acid sequence change, and that protein function may be altered or basically getting rid of. And even though when the sequence is not in the coding region, if it's in the promoter region or enhancer regions or in other regulatory regions, that could impact how the transcription factor binds on that and how that will relate it to the gene regulation process. So we will go through all these different components throughout the semester. So I want to talk a little bit about the technology we use before we have the high throughput sequencing. Okay, very, very quickly. I know that many of you have already learned about that in your molecular biology course. And this is really an essential tool in molecular biology. There is ability to read the basic sequence of DNA molecules. And the most famous ones, and I think both of them won the Nobel Prize. And one is using chemical sequencing, and the other one is using the ddNTP, which is widely referred as the Sanger sequencing. And also that after these sequencing technologies, there is another type of technology called microarray technologies, and we will go through a little bit. And this really generated a very big research discovery throughout the years, especially in the first 10 years of the 21st century. So I'm going to go through this Sanger sequencing a little bit, and for the reason that in the later hours, when I talk about the Illumina sequencing, you can see that Illumina sequencing is really just an extension of the Sanger sequencing, and to a large extent. So the Sanger sequencing reaction is you got this molecule. So we know that we have A, C, G, T, 4 letters in our genome, right? Those are the DNTPs, and then one will be connecting to another, further connecting to another one. So the modification of the DNTP is called DDNTP. So basically, change this OH group into this H, and only these very small changes that this continuous reaction is not going to happen. So meaning that I'm supposed to get on the DNA, and then nobody can go behind me because I'm not having that hand anymore. And so that is the DDNTP part. And when you do the reactions, what you need is really this template DNA, which is the sequence you want to sequence, which you don't know yet. And then you need a primer. It's a very small, short oligo sequence that is reverse complement to a portion of the primer. So you can see this G corresponding to C, C is G, A and T and A, and so on and so forth. And you need a DNA polymerase, which is the enzyme to put another nucleotide, just do this nucleotide extension. And you need a pool of normal nucleotides, which are the DNTPs. And then you need a small proportion of labeled DDNTP. And these DDNTP are the ones that I mentioned is the stoppers, and then they are being labeled. So when the reaction happens is first you got your template, you got this small primer. The primer will be reverse hybridized to this your original template, and then what happens is you got a polymerase and putting ACGT based on this template. So next one is going to be A, and because this is T, and then next one is C, and so on and so forth, right? So what you put here is because of this is a T here, and then the next one is going to be A, and then you can put a normal A, or you can put a labeled A there. Labeled A is going to be the stopper, right? So meaning that when I run the gel through this, so you can see that the first one goes when the A is being labeled as green, and then when you got this stopper going on, and then that is where the reaction cannot further happen. So this molecule is going to be the shortest molecule. So on the gel, it will run the fastest. So you can see this is, you see the green channel, you know that the next one is A. And then for the next one is G, and this is, you can see this and corresponding to C, right? And you see this, you see the green channel, and then you know the next is C, and next is C, C, T, T. And by reading these letters, you're going to get what the sequence is. Yes, Kevin?
So you can count sequence by reading from the smallest DNA sample to the largest DNA sample based on which terminal DD.
Right. Right. Yeah, that is the basic principle. So if you go to the Sanger sequencing very classical textbooks, then that's the way it works. I assume that most of you that and for the at least the biology background knows this process. Okay. So we'll get back into that a little bit when we talk about the general biochemistry protocols of the Illumina sequencers. So next one I want to briefly touch base is the microarray technology. And this technology is actually invented in 1998. And it lasted for more than 10 years in terms of their prime times. And it's very, very smart technology in terms of this is the first time that we can measure the expression level of over 20,000 genes in one single experiment. Right? In the past, when people do this type of gene expression analysis, you can only do maybe one gene at a time using PCR, either qPCR or other type of PCR reactions or Northern blot to begin with, right? So let's go through how this microarray technology works. And we will do some comparison when we talk about RNA sequencing to see why RNA sequencing is better than microarray at this point. So this is the microarray. And you can see this very small size, and you don't really know how big this is. Basically, that's the size of the thumb, our thumb. Okay, it's about this size. And if you look at this cartoon, they have a lot of different small compartments. And each compartment, if you zoom in, it becomes this, right? And then for each individual one, you can see there are many oligos. They are having the exactly same sequence as the gene you want to target, actually reverse complement to that. Okay, so that is the microarray design. You can think of each compartment is targeting one particular gene. Okay, and the sequence is being designed. The sequence of these oligos are being designed to reverse complement to the gene you are trying to target. Okay, so those are the array designs. When we do the experiment, so you can see there is many small oligos being put there. Those are cDNAs or RNA molecules. And they are all having a small red hat there, meaning they're being labeled. There are many different technologies. You can use biotin to label. You can use fluorescence to label. It really doesn't matter. But the bottom line is when this nucleotide found its reverse transcribed, reverse complemented partners, it will hybridize to here, right? So this is the RNA molecule. This is the probe that targets this molecule. You see, they are being hybridized. This is in the process, and this soon will come here. But this particular gene is not a complement to here, so it probably will bind to somewhere else. Okay, so that is basically the hybridization, and the reaction happens. And how we detect the signals is through this. So you can see that for this particular gene, and there's a lot of RNA being transcribed. So we can see a lot of oligos are being hybridized here. And then each individual have this red hat, this labeling there. And then by the end of the day, you are seeing very, very strong signals in these small cellular compartments. And for this particular probe sets, it didn't target, it targets on gene that didn't express. So you can see there is no signal being collected here. And some of them have intermediate ones. So those are very, very basic, very smart microarray technology design. Actually, I have a chip here. And this is the Affymetrix microarray technology. So this small cell compartment is just this little, glass surface you can pass around. I wanted it back, okay, towards the end. All right, so this picture is coming from Affymetrix, and there are several different technologies. And on that, Agilent, Illumina has one, and Nimbogen, some companies are already going away, but they are all based on very similar biochemistry principles. And in terms of the genotyping, there is also a technology, even now we're using a lot, to do the SNP genotyping using microarrays. So if you look at this one, this is really based on the gene, it's trying to measure the gene expression levels. But when you talk about the genotyping, it's called SNP arrays. And what this array does is so you can see that, let's say if we want to target on this particular location, this is a string of gene sequences. And at this location, there are two possible genotypes. One is A, the other is G. Can you see it from the back? Okay, you have to have very good eye. I would not be able to. I can barely see from here. So there is one A, the other one is G. So one is the allele from your father, the other one is from your mother, right? And then what they did is actually they designed probes to target this stretch of sequences. And the only exception is they make 4 probes for each location you want to target. And then you can see all these sequences are the same, and which is being reverse complement to the DNA you want to target. And then the middle part, this is A, C, G, T, all 4 different type of probes. And the way to do that is if you see this particular individual at this location is A and the G, and then when you throw this DNA onto the chip, you know the first row, because this is A, it is not a reverse complement to either A or G. So you can see there is no signal in this first row. Okay. The second row represent this T. So you see that because T is reverse complemented to A, and you are seeing a lot of signal. And the third one is C, and then reverse complement to G, so you're seeing signal. And G doesn't reverse complement to anything, you don't see signal. So by the end of the day, if this is a homozygous, like an AA, and then meaning this is AA, and what you're supposed to see is only for this particular line is being, you'll receive the signal. If it's a heterozygous, these 2 will receive signal, otherwise this will receive the signal. So even now we are using this technology a lot, just for the simple reason of the cost. And if you target on 1 million or 2 million SNPs, the current cost is only about $50 to $100. So it's much cheaper still than the sequencing-based technology. Yes, Kevin?
So these are deployed, so they have 2 copies of each chromosome, but every DNA helix has a complementary base pairing.
Right.
So in this case, like if you look at, wouldn't it be possible, like the strand that has G is also paired to a C, right?
Right.
So wouldn't that C cause G signal?
Wouldn't see what?
Like, are you supposed to like somehow extract like the template strand, or like only get 1 strand of the DNA instead of looking at?
Right, but when you grab a people's blood, right, and there will be 1 million cells. So some of the cells are, I mean, each cell and they have a pair of the DNA, and one is from your father, one is from your mother. And then basically when you do this assays, there are many, many DNA molecules even at one specific location. So some of the DNA molecules will be hybridized to here, others will be hybridized to here.
My question is, like, if you look at your DNA and you have like if you look at someone's genotype,
Right.
you have like a helix that comes from their dad, helix that comes from their mom. Each of these helices have base pairing. Like let's assume that it's like at that position on one strand of A. But within that helix, that A is paired to T, right?
Right.
And then wouldn't that T, if you were to like hybridize, wouldn't that T hybridize
To this A?
Oh, wait.
No, because the remaining part is going to be the same strand of this one, and which you don't do we can go through that after, during the break. It won't happen, yeah. But in general, that is what the microarray is about, right? It's an extremely successful technology, and it's a popular application for gene expression profiling, microarray, and in some modified cases for the splicing analysis. And even now that people are doing that, I think in our university for the clinical laboratory, in the cytogenetics division, they are also using this to detect the DNA copy number variations for the SNP array and for the gene regulation and other things. So there is many problems of microarray, and the major limitation is actually you have to know the sequence before you design the microarray, right? So we have seen those little compartments, and each one has a legal sequence. If you don't know there's a gene there, the chances you are not designing a probe to target that. And also that if even you know the sequence, one cannot fill all of them in a small number of arrays. And for example, if you study human, and I think Affymetrix used to have a product that used 7 arrays to cover the entire human genome. And it's not the whole thing, but it's a major chunk of it. It's very expensive. The data analysis is very messy. The experiment is very time consuming. So it's not really the cost effective way to do that. And then it also have very high noise level because of the cross hybridization and other issues. So we will go a little more detail when we talk about the RNA sequencing part. Okay, so now comes to the next generation sequencing. So what exactly that is? So one can sequence hundreds of millions of short sequences in a single run. And here are the major players. It have the Illumina sequencing, the LAC technology, Roche, Pacific Bio, Complete Genomics. I think at one point, and at the time that people really try to develop the most advanced sequencers, there are over 3 dozens of different companies are working on this problem with many different products. Most of them didn't make it. And but eventually that I think those ideas are being purchased and merged into the current technologies. So when we talk about the next generation sequencing, I want to first talking about a few key words about that. So the first keyword is sequencing, right? So you can sequence the DNA or RNA. In most of the cases, when we sequence the RNA, and we have to convert that into cDNA. So most of the equipments just do the DNA sequencing. But there are now some sequencers, which I will mention later in a little bit. It's a nanopore sequencer. They can directly sequence RNA molecules. Okay. And the second keyword is short reads. And so if, for example, if this is the whole DNA sequence, let's say this is a 1,000 base DNA sequence. It's not like what we imagined initially, sequence from beginning to the end. Rather, it only sequenced the very beginning part of it. And each of this called a read, okay? And how long this read is, it can be initially 35, now up to 150 bases for some technologies, and it could be different longer for other technologies. But general idea is sequence for the short reads. And in some cases that we will sequence from both two ends. This is either called a paired end or made pairs, and which will give those concepts towards the end of today. And the third keyword is really the ultra high throughput. So we are sequencing 5 billion reads in our current the sequencer that's sitting on the campus in our 3 building. And it's really a lot of information to go through. Okay, so now let's go over some of the platforms that what is available for the next generation sequencing now. So the first sequencer is the Illumina sequencing, and it's a leading platform, and it's bridge amplification and sequencing by synthesis, right? I mean, I know you don't know what that means, so we'll get into that after when we talk about the details of the biochemistry. Okay, so if one first gave this lecture in 2011, I would say that Illumina have about 70% of market share, and Solid has a certain percentage, and there's a few other 454 technologies. But now I have to say probably over 90% of the market is Illumina. And this is absolutely a leading platform, and their only competition in this domain is themselves. And in terms of, they have to evolve their technology from the pressure of Wall Street, I think. But other than that, they are really, there's not a lot of choices in terms of this technology. And the second one is solid technology. And this is what we used to have in our school. And if you see, I put a stamp there, it's obsolete now. And it's very, very good sequencer. It's just didn't make the computation. And that is really make all the difference. The third one is called the 454 technology. And this is actually the first ever commercially available next generation sequencing that was put on the market in 2006, is 11 years ago. And there used to be really the cutting edge technology. And for this one, actually, if you see that this is later purchased by Roche, the headquarters is in Fishers, Indiana, which is just US headquarters is here. And a lot of research is being done in this on this campus. And next one is called the Pacific Biosciences, which I will get into a little more detail. And this is, I know that people are talking about next generation sequencing, okay? And then there is another term called third generation sequencing, right? So, for here, I want to explain a little bit about the differences. So the next generation sequencing or the second generation sequencing, what we are talking about for these short read technologies, the third generation is not replacing the second generation. It is not a competition, it's complementary. So there are a few key differences of the two parts. So the first one is single molecule sequencing, okay? And when I get into the technology, you will see that for the Illumina, for example, we need to have amplification before we do the sequencing. So you got one molecule, you don't get enough signal to be sequenced. And you have to amplify that signal to do the sequencing reaction. And for the Pacific Bioscience or third generation sequencing, it's a single molecule. You do not need to do the amplification. So that's the first difference. The second difference is the read length. So I mentioned that the short reads, and that is a feature for the second generation. If we talk about third generation, it can go very, very long reads. And now I think, they are very comfortable by getting the average of 20,000 bases sequences. And in some cases, they can even go to 60,000 bases. It's very, very long sequencing, okay? And those comes with a problem as well. The throughput is not as high as the second generation, and the sequencing error is significant. This is not that accurate. So far that if we really need to do specific certain type of research, we probably need to combine this second generation and third generation together, okay? Just want to make sure that the difference is single molecule and the long arrays. But they are not, say, one is better than the other one. It is complementary to each other. The fifth platform that you are seeing on the market is called M-Torrent or M-Proton technology. And this is a semiconductor-based technology, which we also have in the Center for Medical Genomics. Well, we will see that in our tour. But I will go through the biochemistry later. And but the reason that I mentioned this is it's still very widely used a sequencer, and especially they marketed initially for the clinical laboratories. Okay, they have a huge footprint in China, and I know that they have a lot of marketing efforts in the other side of the world. Initially they have gained a huge momentum in terms of the sequence fast, okay? We're talking about 4 or 5 years ago, when the Illumina sequencer, when you run a run, it will cost you about 2 weeks to do it from beginning to the end. And in many cases, it's not feasible to do that in the clinical setting because patients really need the data coming back immediately. And when they started to develop this technology, they were trying to target on this application, which their assay takes about 2 days, which is a significant shortened period of time. But now other technologies also catch up, so that advantage is gone. And the last one I want to mention is the nanopore technology. Actually, we have tested in the Center for Medical Genomics as well. So you can see this is a person's hand. And this whole thing is a sequencer. Okay, it has a USB port. It can be plugged into your computer. And then after your library prep, you drop your samples into the sequencer, you can see the reads come out. Okay, so it's a really like some sci-fi movie to a certain extent, which I didn't bring in today, but I probably will show you that when we have the tour. It's a very, very small and neat instrumentation. Okay, but these are just the overall view of all the different technologies that are currently available. So let's have a 5 minutes break. We'll come back at 4 o'clock, and then we'll start to get into a little more detail on individual technologies to see why they're sequencing that fast. Okay? 5 minutes break. Don't forget the long page out. The actual hall, and then once you get in, and the meter arrives, there's a wall behind that wall.